Alright, for today's presentation, we're going to take a look at what happens when we're looking at multiple traits. So the reality is that we start discussing genetics by talking about individual traits at a time and seeing what the probability that you'll end up with individual traits is. But the reality is that organisms have more than one chromosome that sexually reproduce, and organisms also have more than one gene on each chromosome. And so this creates a variety of possibilities. So let's take a look at this example here. So here I have a cell, and the cell has its genetic information on chromosomes. And you'll notice that on this cell, we have the information for the capital G allele. Now there's two copies because that's two identical copies of that chromosome. And over here, we have the lowercase g allele. Again, it's two copies because it's two copies of that chromosome. So this is not by itself. It also is here with the Y allele. All right? And so you have big Y, big G, little g. So what happens is during the process of cell division for sexual reproduction, these chromosomes assort. So right here, you have an option where you have big G and big Y together. Right here, you have an option where you have little g and little y together. But down here, where you have the same cell, see, big G, little g, big Y, little y. Here, this time, big G, little y are together, and this time, little g, little g, big Y are together. And so what we've done here is we've created a situation where all possible combinations exist. And so you could end up having gametes with all of these different alleles. So big G and big Y together, little g and little y together, big G and little y together, little g and big Y together. You have gamete possibilities that are any combination. And that's because this process, which is known as meiosis, is what separates these alleles by separating the chromosomes. But meiosis is random. So it's not like you're always going to have the big G and the big Y together. You can have any combination of G and Y. And that leads to multiple possibilities for your alleles. So this adds a layer of complication to understanding the probabilities. So when we want to understand the probabilities, what we're going to do is we're going to take a look at these in terms of how the traits come together. So the first step in looking at the probabilities is to look at the phenotypes. So in our example over here, we have seeds, and the seeds have two different traits. They are either yellow or they are green. And they are either round or they are wrinkled. Okay? And so what we're doing is we're looking at the combination. Are you yellow and round? Are you green and round? Are you yellow and wrinkled, or are you green and wrinkled? So this time we're not just looking, are you yellow or green? We're looking, are you yellow and round, or are you yellow and wrinkled? And so all we're doing is we're adding that layer of a second trait. And since we're looking at more than one trait together, it gives us more possibilities. So what happens here, okay, is the offspring. So here you have, the pure yellow round, the pure green wrinkled, and they breed to create this one, which is mixed. So what you have to do is you have to create the gametes. So here I have A, B, B being crossed with A, B, B. Okay? So what I have to do to actually carry out this cross is to figure out what the possible gametes are. And to do that, we use something called FOIL. I'm sure you've done this in algebra. It's first, outer, inner, last. So what I'm going to do is I'm going to take the first, so A, B, so capital A, capital B, so the dominant of A and the dominant of B, and you'll see that that possibility is placed here at the top of my Punnett square. Then I'm going to do the outer, so A, capital A, and lowercase b. So the dominant of the A and the recessive of the B. And you'll see that that one is right here. Then I'm going to do inner, so the 2 on the inside, so lowercase a and capital B. All right. And I'm going to place that here, so that's the recessive of the A and the dominant of the B. And then I'm going to do last, so the capital A, or I'm sorry, the lowercase a and the lowercase b. And you'll see that that one goes there. Now the reason we use FOIL is because we just want to make sure we put all the possibilities there. It's not something you have to do, it's just a tool we use to make sure we have hit all the things that could be. All right? Now, in this case, you'll see that the parents are both the same, so the gametes on the top are going to be similar, not similar, they're identical, to the gametes along the side. Now, just like before, what we're doing is we are combining them to see what 2 A's we get and what 2 B's we get. So one of our first options is to see where we can find the yellow and the round together. So right here I have capital A and capital A, capital B, and capital B. That is the yellow phenotype and the round phenotype. Here I have capital A, capital B, lowercase b. That is also yellow and round. Here I have capital A, lowercase a, capital B, again yellow and round. And here I have capital A, lowercase b, a, capital B, lowercase b, again yellow and round. And then the same thing is true here, 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 and here. So there's actually 16 boxes here. And out of the 16 boxes, 9 are yellow and round. So now I'm going to look for green and round. Okay? So green and round. Little a, big B, little b. Makes sense. That's green and round. Little a, big B. All right, so that's still green and round. Little a, big B, little b. That's green and round. So out of all of these, 3 are green and round. Next up, I'm curious about yellow and wrinkled. So here I have homozygous dominant for A and homozygous recessive for B, heterozygous for A, homozygous recessive for B, heterozygous for A, homozygous recessive for B. So again, I have a situation where 3 are yellow and wrinkled. And then the last possibility, which we see right here, is wrinkled and green, and that is little a, little b, and so that only happens in 1 out of the 16. All right? So this right here is known as a dihybrid cross. So all we're doing is we're looking at 2 traits at a time. The reality is that we really don't use this that much. Instead, what we like to do is what we call multiplicative probability. Okay, so if we go back to the example we just did, all right, and you have 9 to 3 to 3 to 1, okay, your chance of being green and wrinkled, okay, your chance of being green and wrinkled is 1 out of 16. Okay, now we did that by doing a dihybrid cross and seeing all the ways that the gametes can combine. But instead what I could do is this. I could do my chance of being green. So over here we used A for color. So this was my parent, and this was my parent for A. Okay, that was my parent for A. So right here, my chance of being green is 1 out of 4. Okay, ignore this for a second. Let's instead look at wrinkled, all right? So my option for wrinkled was B, so that was one parent who was heterozygous for wrinkled. And here's my other parent who was heterozygous for wrinkled. Okay. And when I combine them, I end up with homozygous dominant, heterozygous, heterozygous, and homozygous recessive. And so my chance of being wrinkled is also 1 out of 4. So my possibility of being green is 1 out of 4. My possibility of being wrinkled is 1 out of 4. So when I want to use multiplicative probability, what I'm going to do is I'm going to take my possibility of being the one thing, because these are independent, and I'm going to multiply it by my possibility of being the other. Well, when you multiply fractions, 1 times 1 is 1, 4 times 4 is 16. So I have a 1 out of 16 chance of being green and wrinkled based on multiplicative probability. You'll notice that that is the same as doing a dihybrid cross. The reason that multiplicative probability is so useful is that you can do this for any number of traits. So if I want to do 2 traits, I do it this way. I want to do 3, I do it this way. I want to do 4, I do it this way. And the reason this works is because of something known as the law of independent assortment, which basically just says that our traits sort randomly, just like we saw on that picture of the chromosomes. And one of the big things that makes sure that this actually happens is a process called crossing over. Because crossing over actually prevents most of our genes from being linked. And because most of our genes are not linked, you know, the gene for eye color is not connected to the gene for hair color. That's why you could have blue eyes and blonde hair, blue eyes and brown hair. Whereas if they were linked, you might not be able to have one without the other. There are some genes that are linked because they tend to be real close to the centromere, but thankfully it's not many. And so these probabilities play out for most of our traits. All right, that's it for this presentation.